Welcome to Capsugel Dosage Form Solutions Bioavailability Enhancement webinar series. Today's webinar will address lipid-based formulations, scale-up and manufacturing aspects, and will be hosted by myself, Eduardo Jewell, a Senior Manager in Pharmaceutical Business Development, and my colleague Stephen Brown, Manager Director of our NCAP division. Our latest webinar highlighted new chemical entity challenges related to poor solubility. It also outlined the advantages of this technology that go well beyond bioavailability enhancement into food effect decrease and reduction in variability. Importantly, we reviewed case studies that describe that our approach to take a drug from feasibility to lead formulation dosing in animals in 12 weeks. As a reminder, all of our Capsugel webinar series are available on our website. In the agenda today, we have lipid-based formulations, overview and applications. And even though Capsugel offers soft gelatin capsule development and manufacturing, today's talk will be focused on liquid-filled hard capsules, from formulation characterization into manufacturing and process transfer considerations, all while covering a few case studies that address process transfer and scale-up studies, from Vancomycin, a well-known thermosetting liquid fill in gelatin capsules, all the way into product A, and liquid fill into HPMC capsules. Last but not least, we will have a 10-minute session to address any concerns you may have. Capsugel Dosage Form Solutions business model consists of three major steps: design, develop, and manufacture, from a target product profile into finished products. And all of these three steps are based on strong foundations of science and engineering, as our multidisciplinary teams engage with client teams, technology breadth and selection, on which we'll be expanding in just another slide, rapid advancement in quality by design, as well as Capsugel being your partner of choice through a very collaborative approach. And even though our webinar today will concentrate on lipids, i.e. oils, co-surfactants, surfactants, and co-solvents, also formulated as self-emulsifying and self-microemulsifying drug delivery systems, our technology offer is much wider, from bioavailability enhancement through amorphous systems such as spray-dried dispersions or hot melt extrusions, into nanocrystals, into modified and targeted release, all the way into specialized applications such as multiparticulates, inhalation formulations, abuse deterrence, and taste masking approaches. Technology selection, or the process through which we identify the ideal technology for a client's formulation program, is, as we said before, a process that's based on modeling that takes into account key physical, chemical, and biopharmaceutical properties of the compounds, such as solubility, log P, melting point, but also efflux or potential metabolism. Now, this process obviously relies on years of experience in formulation and product development. Now let's talk about lipids a little bit more. Lipid formulations are well tolerated and absorbed and can increase drug solubilization in the intestinal lumen through dispersion and/or digestion, but also inhibit enteroside-based efflux transfer mechanisms mediated by BGP or BCRP receptors and/or by intracellular metabolic processes, often mediated by, say, the P450 subenzyme families. In addition, there's growing evidence that shows that lipid formulations can also redirect the absorption pathway from the portal vein into the lymphatic route. In order to bring clarity to the lipid formulation realm, Colin Powell from Monash University introduced the LFCS, or Lipid Formulation Classification System, in 2003. The LFCS defines formulation types that are based on composition, but critically, on the risk of drug precipitation following dispersion and digestion, two key criteria that characterize the performance of these formulations that range from oils to self-microemulsifying drug delivery systems, and also from liquids to solids. And as mentioned before, Whereas the primary application of these technologies is bioavailability enhancement, lipids have also been used to contain high potency or low dose drugs, provide abuse deterrence properties by suspension of the drug in a non-crushable soft matrix, all the way into 505(b)(2) and other life cycle management strategies. Lipid-based formulations have commercial precedence, as a number of products have reached the market in the past in the shape of soft gelatin capsules, but also hard capsules. In addition, there's a number of new chemical entities that are currently at different stages of development utilizing these versatile technologies here at Capsugel. And one of the key advantages of this dosage form is precisely versatility, as hard capsules can be filled with liquids, and that can be liquid actives, but also liquid formulations such as solutions and suspensions, SEDs and SMEDs, all the way into solid and semi-solid formulations that can be, again, solutions or suspensions. And given this wide range of physical states and compositions, products should be well characterized and understood prior to scale up and manufacturing. And that's precisely where our next section is going to cover. Scale up through formulation characterization. One of the first things we look at when identifying a lead formulation is compatibility. And whether they are made of gelatin, HPMC, or purulent, most hard capsules solely contain polymer and water, which acts as a plasticizer. In order to develop a robust dosage form, then, the interaction between the encapsulated materials, capsule shell, and the environment should be fully understood. For those of you who use our capsules, you will have noticed the label on our boxes. Capsules, in particular gelatin, should be stored at 15 to 25 degrees C, 35 to 65% relative humidity, which in the case of gelatin is the ideal range for capsule shells to retain 10 to 18% water in the shell. Outside of this range, shells may soften or even become brittle and obviously compromise the quality and the robustness of your product. In order to present the properties of the shell, long-term compatibility should be predicted through careful evaluation of hygroscopicity, or the moisture exchanges between the formulation, the shell, and the environment, loss and drying and its potential impact on mechanical resistance, as well as dissolution, which can be used to predict potential cross-linking. Formulations may further be classified according to the rheological properties, especially in preparation to the filling process. From Newtonian liquids or thixotropic matrices that may be filled at room temperature, into semi-solid or thermosetting matrices that may exhibit Newtonian or non-Newtonian, shear thinning being the most common when filled at high temperature. In any case, especially thixotropic or thermosetting vehicles need to be filled at high temperature, up to 70 degrees, in order to reduce the viscosity of the system. And whereas liquid formulations can be easily characterized through density measurements, as most filling operations are volumetric, semi-solid formulations require additional understanding, especially at higher temperatures. The melting profile provides guidance to define the mixing and filling temperatures. Hold time studies confirm stability at simulated mixing and filling temperatures, especially during extended periods of time. And then the viscosity range will guide the identification of mixing, the device, and the speed to filling, the nozzle types, and the diameter parameters. In this particular case study, the melting point of this formulation was determined to be 35 or 39 depending on the cycle. However, the end of the melting cycle in either case was determined to be over 40 degrees, for which a filling temperature of 50 was identified. Now, when conducting thermoviscosity measurements or thermorheology measurements, we determined that the viscosity of the system was extremely low, well below 50 centipoise, and closer to the 20 to 30 centipoise range, which requires obviously careful selection of the nozzle, as key to the sealing process is actually the filling. We want to avoid having splashes during the filling, having dripping or potential bridges that could contaminate the bushes around in the capsule filling machine, as well as potentially the sealing area in the capsules. Other mixing and filling parameters may need to be implemented following determination of rheological properties of the system. In this particular example, a high viscosity and viscoelastic system required additional controls, such as during the mixing step, robust temperature monitoring, scraper blades to ensure proper heat distribution, mixing, and homogeneity, an isolated or heated recirculating tubing to maintain an ideal temperature window. On the filling side of things, this process required heated nozzles as well as nozzle dipping to make sure we were able to fill the formulation from the bottom of the capsule all the way up and avoid potential spills. If a solution of the active ingredient cannot be achieved, then a dispersion or a suspension can be formulated. Ideally, the system should have low hygroscopicity, good dispersion stability to avoid segregation or sedimentation during filling and storage, and thixotropic systems in particular, which have an apparent viscosity which is suitable for filling while maintaining dispersion homogeneity, and restructure post-filling with an increase in apparent viscosity, providing improved stability and obviously reduced leak prior to the sealing process. That's it for my part. Let me now hand over to Stephen Brown, who's Managing Director of our NCAP Drug Delivery Unit in Edinburgh. Stephen will be covering today all of the manufacturing aspects from API processing to packaging. Stephen?
Okay, thank you very much, Ed, for the introduction. As Ed said, I'm Managing Director of the NCAP Drug Delivery site based in Edinburgh. And NCAP as a company has been involved in liquid-filled hard capsule manufacture for approximately 25 years. And what I'd like to do this afternoon is to take you through some of the steps that are involved in liquid-filled hard capsule manufacturing, and maybe go through some examples of products that we have been involved in the development of or scale up from our clients into commercial manufacture. So this schematic shows the liquid-filled hard capsule manufacturing process, which starts off with formulation preparation, and this can be from a very simple formulation of actually just a liquid API, or it could be a solid API, which is then incorporated into a liquid excipient, and these two would be mixed together to produce either a solution or a suspension. On some occasions, the excipient is a thermosetting vehicle which needs to be heated to become a molten liquid before incorporation of the solid API. And we can, of course, have more complicated formulations that contain multiple excipients. So once those materials are all together in the mixing vessel, there's a mixing process, and as part of that mixing process, occasionally we can introduce air into the system. So these mixing tanks that we have need to be able to draw vacuum to degas the mix. The mixing tank is then transferred to the filling machine, where the liquid product mix would be transferred into the hopper of the filling machine, and the capsules filled and closed. The filled capsules are then transferred to a sealing equipment. This can be two different routes. It can be banded, sealed by banding, which is something that NCAP have most experience with. And this is where the capsule is sealed by placing a band around the join between the cap and the body. Alternative possibility is the proprietary Capsugel LEMS technology, which is liquid encapsulation microspray system. In this technology, a very fine spray is sprayed around the seal between the join between the cap and the body, which effectively fuses the two parts of the capsule and performs a very good seal. In our experience, both technologies work extremely well from a technical perspective, producing leak-free capsules. In certain processes, routes here, the product will actually transfer from stage 4 sealing directly to weight sorting and metal detection, and then on to final commercial packaging. For some of those products which could be prone to leaking, or where there's a truly mobile liquid inside the shell, then in this instance capsules are trayed and placed in a vacuum chamber. The vacuum testing would then be conducted either at ambient temperature or occasionally at elevated temperature. Following the vacuum testing or the vacuum cycle, capsules are inspected and then would follow through normally into the weight sorting and metal detection process as before. And on this slide here you see also the option of capsule coating. This is something that we've been involved with for a number of years. Primarily functional coating of capsules. So this is enteric coatings, erodible coatings, coatings that would allow the capsule to pass through the stomach and into the small intestine, or indeed targeting to the colon. And we'll talk a bit more about those in the next few slides. This slide shows liquid-filled hard capsule processing in a more schematic way and looking at the individual unit processes. And I have split this up here into process steps which are low or challenge in terms of scale up, and those processes which are can be more challenging. And the more challenging ones would be where we have to occasionally have powders or APIs that need to be processed prior to the mixing step. So milling of materials to produce particles, a different particle size. The mixing step itself can be more difficult to scale up, and also the coating process. But these other unit processes which are on the slide are generally relatively low in terms of scale-up challenge. And for example, the filling machine, if we have a formulation and a process with which we can fill a few thousand capsules on a commercial filling machine, then there's absolutely no reason why that same formulation process cannot be used to produce millions of capsules. And in the lower part of the slide we have demonstrated here two scales that we operate at. So in the early stages of development and early clinical manufacture, we would be working at a scale of materials of 100 grams to 5 kilos, and generally using bench scale equipment, bench scale filling machines, bench scale sealing machines, and bench scale coating machines. At the upper end of this limit, around the 5 kilo scale, we would usually be aiming to take the process, the formulation and the process onto actually commercial scale equipment. Our philosophy has always been to get our formulations and processes onto commercial scale equipment as soon as possible in the development phase. As this then allows us to demonstrate formulations and products that will be subsequently easily scalable to commercial. In the second bar, we're talking about larger scale clinical supplies and commercial, where the normal scale that we would operate at here would be in the 5 to 200 kilo range for mixing and filling, and 10 to 150 kilograms for when we get to coating capsules. For all new products that we would bring on site here at NCAP, the process is essentially the same. So in this particular example, if we are developing a product for our client this is the standard process that we would go through. We would initially do pre-formulation, where we are aiming to confirm the compatibility of the active ingredient with the various formulation excipients that we might use in formulation, and also how the compatibility of the active and the excipients with the hard shell capsules, particularly with gelatin, HPMC. And as we conduct these studies, we are also looking at factors that may affect the stability or performance of the active and or the excipients. So we'll be looking at temperature, how that might affect the stability of the drug. So we'll be conducting temperature hold and temperature cycling studies, for example. And in terms of the overall process here, moving through from pre-formulation, if we are developing self-emulsifying systems, then we would be looking at the emulsion and dispersion characteristics and digestion characteristics of those formulations in vitro, before we then move on to defining prototype formulations, which would be further characterized for in vitro for dissolution and stability. And we would have an optimization loop at this stage where if formulations developed did not meet the criteria that we had set on in vitro characterization, then there would be a loop back into further formulation development. Eventually we would move through to technical manufacture and stability of those batches, and some of those batches would then be normally taken through to in vivo pharmacokinetic studies. And this, on the right here on the pictures, you would see the sort of equipment that we would normally use. So here we have bench scale high bar capsule filling machine and a quality bench top capsule bander. So this would be the scale that we would operate at normally for in this stage. Occasionally, if a client comes to us with formulations that they have already developed, and this happens quite frequently, where clients will have done some formulation work and they are coming to us for process development and scale up, we would almost always evaluate those formulations at this scale before we would actually progress through to commercial scale equipment. This next slide shows, the previous slide I showed you the equipment, bench scale equipment that we use for filling and banding. This is a CFS 1500, which is a combined capsule filling and sealing system developed by Capsugel, and this is bench scale type equipment, allowing us to operate up to 1500 capsules per hour, and this is really ideal for feasibility studies and for the determination of mixing, filling and sealing parameters. And we have very good experience with scaling up to commercial scale equipment, to the LEMS equipment from the CFS 1500. So as we move then on from the early development phase through to the first start of scaling up, in terms of mixing here we have basically equipment which is around 20 kilogram scale, and essentially all of the mixers that we would have, even as we scale up from this level, would have very similar capability in terms of they would be jacketed mixing vessels, because we need to, many of our excipients require heating and temperature control. Almost always we would have incorporate the active into the mix using high shear, but also having paddle mixing capability. And on the right-hand side, we've got some information here on what the scale-up challenges are, and it's really dependent on the type of the formulation. So if we have a liquid API or a drug in solution in liquid fill excipients, then the scale-up challenge is relatively low. And we really need to only define and control the agitation and mixing and temperature that we need to actually get a good solution or a homogeneous mix. Suspensions are slightly more difficult and need, there are further aspects here that we need to consider. For example, we need to achieve and maintain suspension stability both during the processing and the product storage once filled into capsules. So during the mixing phase, we will want to achieve good homogeneity of the active within the suspension, and we will then want to have good suspension homogeneity maintained while the mixed product is held either in the mixing tank or on transfer into the hopper of the filling machine. So the things that we need to consider are potential for settling of solid material. So we're looking at particle size. Because these will tend to be mixed systems, in other words, there will be some drug possibly in solution, some drug in suspension, we need to consider the possibility at least of particles of recrystallization or changes in crystallinity with time. And these are more of a theoretical issue. In all of the time that the projects that we've been involved with here at NCAP, over 200 projects, this particular situation has really only arisen on no more than 2 or 3 times. So it's a theoretical consideration and something that needs to be evaluated, but not a major concern. So on the right-hand side of the table, we see the things that we need to control are control API particle size if we're generating suspension, because that can be important for stability and also for dissolution. And we need to formulate for processing and stability. So we may often add additional excipients, thixotropic agents or thickening agents to actually modify the suspension processing capability. And as we move on from the 20 kilogram scale to the larger scale mixers, we have in these pictures 2 examples of some of the larger scale machines that we have. And these are essentially very similar to the smaller scale in terms of capability. So in other words, they're temperature controlled, jacketed, both have high shear capability. The Skirman on the left has high shear and paddle mixing integral to the mixer. The Gusti mixer on the right has actually its high shear as an external high shear. The interesting point on the Skirman on the left is we'll see the pipe work, which allows recirculation of mix from the tank back into the tank at the top of the mixer. And this is a novel innovation that we've developed to allow powder addition to the mixing tank without actually opening the tank. So this is a very useful way of adding powdered materials into the mix without causing material blooming around the upper surface of the mixer, which can be very important. So we move on now to the filling process, and the filling on a standard filling machine is a relatively straightforward and simple process. We have at the top of the schematic here a jacketed tank which holds the molten liquid or the liquid mix of the product that we're going to fill. Just below that there's a dosing pump block. So this will deliver the drug product through the filling needles or the filling nozzles directly into the empty capsule body with the cap removed. So as the dosing needles insert into the capsule, the product is dosed and then the needle is retracted. And the issues that can be problematic here are dripping and tailing at the filling nozzle. So the things that we need to control here particularly are process parameters such as nozzle selection, filling speed and drawback, and of course the actual formulation itself. So with these control parameters we can define the right formulation that will fill properly on the filling machine as long as we've selected the right nozzles, the right filling speed and drawback. The other important factor, particularly for thermosetting materials, is that we need to operate at a temperature that allows the filling machine to maintain product temperature all the way through from the mixing hoppers on the filling machine right through the dosing pump block until delivery of the material into the capsule shell. And now some discussion about the sealing, and we've already discussed the two potential routes of sealing, banding, which NCAP have much more experience with. And the issues really here are around transfer of capsules from the filling machine to the rollers and to the slats of the banding machine. So transfer across the main body of the banding machine, where the band is applied by discs, which pick up the banding solution from a reservoir and apply it around the middle of the capsule between the cap and the body. So there are a number of things that we can control in the banding process. We can modify the formulation of the sealing solution itself. In the banding composition, we can adjust the discs. We can have different disc formats and speeds. We can adjust the height of the disc and the temperature of the drying chamber and speed and alignment of the machine. So all of these things are aspects of the process that we can modify. In terms of lens, the microspray sealing technology, similarly we have the challenges are transfer from the filling to the sealing station, and one of the advantages of the lens technology is the vertical transfer. So in this particular instance, the capsules are held in a vertical position while they on transfer from the filling machine to the sealing station. This obviously reduces the risk of potential leakage. And in our experience, as I said before, both of these technologies work extremely well with very low leak rates. My experience would be that for very low viscosity solutions, the lens would be a better choice than banding. Next, we want to discuss a little bit about capsule coating. Capsule coating of using these functional coatings that we talked about earlier is essentially the challenges on scale up here are identical to what we would have for tablets or particle coating for that matter. The one area that might be slightly more problematic is the possibility of overspraying. So particularly if we're spraying with aqueous coating solutions, then there is a potential that capsules of gelatin or HPMC would be more susceptible to aqueous damage. So it's very important to get the spray rate and the spray conditions right so as not to overspray. It's also very important to get good coating uniformity and good capsule mixing within the capsule coater itself. And there are a number of things that we can use here to control, which are very well known in the industry: spray rate, airflow, inlet temperature, atomization pressure, and drum speed. And within our Bend research organization in Oregon, we have a lot of experience in coating tablets and particles, and a number of technologies and techniques that allow us to really define the correct parameters. For example, we've got spray pattern measurement and droplet size across the spray, so we can look at different parameters and how they affect the droplet particle size and the spray pattern, and then select the most appropriate for use in the coating equipment. In terms of all of our development projects, we always keep quality by design at the forefront of our thinking. And although the liquid fill process is relatively straightforward and often can just be the combination of an API with a liquid and filling into capsules, which sounds, very straightforward, there are a number of parameters that we can consider in terms of material attributes, the capsules themselves, the various parameters that we've already discussed around mixing, filling, banding, coating. And as we move through the development process from smaller scale to larger scale, we're always conducting a risk assessment of the process and the formulation and the process. And we will try to design experiments at the edge of failure for material attributes and process parameters to define the design space that we can operate in. And obviously with a view to the quality, critical control attributes and control strategy that we can put in place to achieve a quality product. So I'm now going to move on to a few examples, and NCAP in our 25-year history have approximately 28 to 30 marketing authorizations, so different products in commercial manufacture, and over 100 development projects that we've taken through into clinical manufacture. And I've selected 4 products here which kind of demonstrate really the scale, the levels of scale that we can operate at, and also products with different characteristics. So for example, product A is a highly potent product, which is an oncology product. It is a true liquid-filled and banded product. It's in HPMC, and the reason that product is in a liquid-filled format is for bioavailability enhancement, and we've effectively scaled that up to a commercial scale of 100,000 capsules per batch. Product E is a thermosetting semi-solid matrix, banded product. The reason this product is in the liquid format really is through processability. So a number of the excipients and components lend themselves very well to the liquid fill technology. And this particular product, we're manufacturing up to 1 million capsules a day. So we'll go through these just one by one. Vancomycin is as I said, is a thermosetting fill. This is one of the original products that was developed in the liquid fill format by Eli Lilly back in 1988. You can see the paper here. And this was, at that time, a new approach for vancomycin. Vancomycin is a material, is an antibiotic, which is supplied as a lyophilized powder due to stability. The material is very hygroscopic. It's hydrolytically unstable and it's oxidation sensitive. And the original developers of the oral dosage form found that they were unable to develop a powder-filled or tablet formulation that was gave a commercial shelf life. So they were looking to develop, they decided to look at semi-solid thermosetting fills, but they were looking to have a product with a particular rapid, immediate dissolution profile. They also needed stability based on the characteristics of the compound, so they needed to be careful about the temperature and moisture sensitivity. They needed a product with the correct rheology and flow characteristics, and they evaluated a number of liquid fill semi-solid matrices, and you see some graphs here that they explored with PEG 4000, PEG 6000 at various temperatures that could potentially be used for filling, and they were exploring what the behavior of the materials were in those excipients, and then tried to identify formulations that would then give good stable product, but also process on a filling machine. And the process of manufacturing of vancomycin is relatively straightforward. The PEG is melted, the drug is incorporated into the melted PEG with high shear. Obviously, the entire mix is being held at temperature here of 65 to 70 degrees, and this is maintained all the way through the manufacturing process from the mixing tank to the hoppers of the filling machine. And there also is the removal of entrapped air, so the degassing step. And then capsules are filled and metal checked and check weighed. And you can see here that, in terms of these parameters, in-process assay, in-process fill weight, QC fill weight, we have a very consistent levels of for each of these, with very low standard deviations of less than 1%. So a very effective process and reproducible. The second product I want to talk briefly about is another thermosetting fill. This is a product that we transferred from a client, and the client provides us a pre-mixed drum of 200 kilos of product. And the process that is involved is basically heating the drum, degassing of the mixture, and then filling of capsules. And normally in these situations we would heat the entire contents of the drum using a drum heater. This can be quite a lengthy process and often would be done overnight. Because of the requirements to increase our manufacturing speed and capability, we actually designed for this product a floating barrel heater, which was a novel design. So there's a heater which sits on top of the mix, and as the mix melts and is drawn off into a degassing chamber and subsequently onto the filling machine, then the heater gradually moves down the barrel as the material melts. So this is a very fast, efficient process and allows us to fill 1 million capsules a day. And we've filled so far 112 million capsules filled. And you can see from the graph here that the fill weight control is exceptionally good, plus or minus 1.25% of the target. And this is across 20 batches of 20 million. This next slide shows another thermosetting fill. In this instance, we have a liquid API, which is in a hot melt excipient. And we, again, transferred this process from a client. We used 7 batches to develop the process on the equipment train and complete the process validation. Up to 28 million capsules have been filled, and with this process, we were able to fill about 550,000 capsules per day. The interesting point here is that mix homogeneity is a critical consideration for this product, so we need to ensure a very good mix of the liquid API with the hot melt excipient. So again, we've developed some very interesting automatic process cycle here, where the product needs to be vacuumed to degas after the very intensive mixing, and then prior to pumping over to the filling hoppers, the tank has to be returned to ambient pressure with nitrogen. And I'm going to show you some the actual setup on the equipment on the next slide. On this slide here, we just see the across a range again of 20 batches. We see excellent fill weight control with standard deviations less than 1% generally. And this shows the setup that we have. So we have the filling machine on the right with the filling hoppers, and we have level sensors in the filling hoppers. So whenever the product is filled down to, and the hoppers are 25% full, there's a signal sent to a PLC controller which changes which works in on the valve on the GUSTI, which basically returns the tank to ambient pressure and allows automatic pumping of liquid product through the transfer pipe work back into the hoppers. When the hoppers filled, then it switches the system back to vacuum mode. So with continuous mixing and recirculation all the time. So this is basically an automatic process, so the operators can be focused on the filling machine while the mixing and degassing all occurs automatically. And the last product example here is the product A in HPMC capsules. So this was a product developed by the client, 3-dose strengths. As we transferred into NCAP, we did a number of processing studies looking at shear rates, filling speed, nozzle size, operating temperatures. And this was initially done at batch sizes of 2 kilos, but on commercial equipment. So this really is the lower limit of batch size that can be handled on commercial equipment. But this allowed us then to do process scale up from 2,000 capsules to 25,000 to 50 and eventually to 100. And this is a particularly challenging product because of the low capsule size and low fill weight, particularly for the banding process here with capsule transfers across the banding machine. But you can see that across 3 campaigns, there was a lot of banding development work being conducted in the first campaign across 8 batches, with an overall yield of just over 70%. But as we increase and modify the processing as we moved across the campaigns, and eventually we're up to yields of an average of over 95%, which is very good for this particular process. As I said, this was a high containment product, so this slide gives you an overview of the high containment area which we have at the NCAP facility. So this is a completely dedicated area to highly potent actives, own separate air handling, change control, so people access at the bottom of the area through the green area, and materials transfer through the top, then into a dispensary area, which is a contained room, which contains the Hosokawa isolator for dispensing the API. The API is then automatically transferred through into the next room, the mixing room, where we have the Dynex mixer, and then automatic transfer of the compounded liquid material through into filling 2, as on the diagram where we have a Bosch filling machine and bander, S100 bander. So in summary, lipids and liquid-based formulations are very versatile in addressing a number of issues. Primarily, bioavailability is the one that people are most aware of, but it's also very useful for low-dose products, for highly potent products. There are many examples of products which we can actually improve the stability of the API using thermosetting or semi-solid matrices and other formulation challenges. Careful system characterization is a must prior to initiating manufacturing and formulation work. And while certain aspects of the process that we've discussed today, for example, the milling process, the mixing process, and the coating can have its challenges, overall, scale-up and manufacturing of liquid-filled hard capsules is a very straightforward, and we've demonstrated a very effective manufacturing technology. There are multiple polymer options in terms of hard capsule possibilities, gelatin, HPMC, and pullulan. And capsule sealing in our hands with either banding or lens is a very highly effective and proven technologies that can utilize shells, again, from both gelatin and the HPMC. The technology also, the liquid-filled hard capsule technology also lends itself to other formulation possibilities such as capsule and capsule technology for dual release, and as we've talked about today, colonic delivery to the small intestine, and also abuse deterrent formulations. So the last slide in the on this part of the presentation covers the Capsugel DFS product development and manufacturing sites, and we have 3 primary pharmaceutical development manufacturing sites in Bend, Oregon, where we specialize in amorphous dispersions, controlled release, and biotherapeutics. We have the NCAP drug delivery site in Edinburgh that I've been talking about today, which specializes in liquid-filled hard capsule, high potency, and controlled substances. Strasbourg-Ploermel in France, where the platform focus is soft gels, liquid-filled hard capsules, with a specialization in highly potent products and hormones. And then we have 3 health and nutrition sites in Japan, France, and Greenwood, USA. And overall, we have over 20 years experience, greater than 3 billion commercial units produced annually across our sites, and the ability to handle highly potent products up to OEB 4 or 5. So with that, I thank you for your attention, and I'm going to hand over back to Ed for questions.
Thanks, Stephen. Thanks. This concludes our presentations today. We have some time to address any concerns you may have. We already have some questions coming in. I have a question already here, and we actually encourage you to keep them coming. Stephen, is there a way to introduce nitrogen into the mixing chamber?
Yes, for all of these mixing tanks, it's possible to obviously to vacuum, to have a vacuum line on the chamber, and also then to reintroduce either air or nitrogen depending on the product requirements.
Great, great. Thanks for that. I have another question coming in. Is high potency API formulation better suited to liquid fill or soft gelatin capsule formulations based on our experience? Want to take that as well, Stephen?
Yeah, I mean, I think the answer to that really is that for these types of formulations where the API can be incorporated into a liquid prior to filling either into a liquid-filled hard gel or a soft gel, really both formats are ideally suited to highly potent actives. And we've had growing interest in both formats for these types of compounds, both liquid-fill and soft gels.
Great. Jasmine, thanks for the compliments. We also had a great time. Cheryl, thanks for your interest in abuse deterrence. Here's another question coming in on whether lipid formulations are suitable for opioids or other controlled substances when abuse is a formulation factor.
Yeah, this is an interesting area for that we've seen increasing interest in terms of abuse deterrent formulations for opioids and other products that are liable to abuse. And again, the semi-solid nature of some of the formulations that we can develop can make these formulations more resistant to the types of abuse to inadvertent release of the active through chewing or dissolution. So, yeah, we find increasing interest and have a number of strategies that we have used to basically reduce the ease with which people can extract the compounds from these types of formulations.
Thanks, Stephen, and thanks again to the participants for the other questions that keep coming. I have another one here on the choice between banding or sealing. Is there a cost-associated factor there?
Well, so most of our experience over the last 25 years at NCAP has been banding. More recently, since our involvement with Capsugel, we now have experience with the LEMS technology as well. In terms of processing, we found that both routes work extremely well. The choice between the two really can often come down to customer choice and branding. Banded capsules does give the opportunity to have a visible seal, which can be important for some products, particularly OTC products. In terms of the types of formulations, there's really not much to choose between the two technologies, although I would say that for particularly low viscosity formulations, then I would say that the LEMS technology would be the one of choice.
Thanks, Stephen. And from the Capsugel perspective, I guess that's our experience as well. The LEMS provides an elegant alternative to banding, as the sealing is now within the capsule. It's invisible to the external eye, so it's a matter of customer preference. But when it comes to cost, I mean, both forms are fairly competitive, so there's definitely no difference there. I have another question from Jasmine. Do we have any experience with proteins, peptides, or large molecular molecules using liquid capsule filling?
Yes, we do. Really the interest is primarily in the delivery of peptides and proteins to within the GI tract, and not necessarily for systemic availability. So we're looking for peptides that might be interacting with receptors in the GI tract, primarily the colon, although we do have also had some experience with a few formulations where, particularly lower molecular weight peptides, where we are moving products from injectable to oral delivery. So we have had experience with both.
Absolutely, and I have one more question here on what polymer systems or what polymers do you use for coating? I guess I can start off and then pass it on for you to you, Stephen. I guess we can say we use pretty much the same polymers that are used for standard tablet coating. Yes,
exactly the same polymers that would normally be used for tablets are routinely used by us for capsule coating. These can be pH polymer systems. Erodible coats are also quite often used, so we can, which would provide more of a timed release through the GI tract, and we also have some experience with microbial colonic target systems.
Okay, I have another question here on scale up, whether characterizing the formulation and doing some feasibility runs is really everything it takes to move from, I would say, the small scale to the large scale. I can start off and then hand over again, Stephen. I think that is correct. I mean, we went through the slides back in the presentation, and we discussed how obviously fundamental understanding the formulation, its properties, and more specifically its interaction with the capsule shell, be that a gelatin, HPMC, or pure LEM capsule, is key, of course, is critical to somehow predict the compatibility or the robustness of the product. Then it's all about understanding the rheological property, the density in the case of a liquid, the melting profile, rheological properties of the API. But then I would say, Stephen, it's really whether we fill 100 or 100,000 capsules, the process is pretty much the same, so scale up is, for the most part, very straightforward.
Yeah, I would agree with that. But, as I said, if you develop, the key things are the formulation itself and the process, and if you have those two things optimized, then the scale-up process, certainly for the filling part of the operation, is very straightforward.
Great. We are running out of time, I believe. still a couple of questions, but we can cover that separately. I guess this concludes our webinar today. Thanks again for your participation and for all of your questions. I again encourage you to view our other webinars, especially in the Bioavailability Enhancement series. All of them are available at our website at capsugel.com or at bendresearch.com. And also we encourage to reach out to Dosage Form Solutions via email in case you have any other questions or wish to discuss any specific projects at DFS, as Dosage Form Solutions, DFS inquiry at capsugel.com. With that, thanks everyone again, and we'll see you soon. Bye-bye now.